So I'd like to talk to you about how microtubules are detyrosinated. And I'm sure that a lot of you do know about microtubules. They're key components of the cytoskeleton, that they define the cell shape, they help the cells move, and so on. They're also involved as railways for organelle support by different types of enzymatic complexes that transport vesicles and so on. And they're also very heavily involved in mitosis for moving the sister chromatids in the opposing poles. And something a bit more of a smaller thing is that, but important thing if you're in a cancer institute at least, is that they're the target of the taxane class of chemotherapeutics that are being quite widely used in the clinic for a variety of reasons. So one thing that is lesser known about the microtubules is the so-called tubulin code, which is a series of post-translational modifications that microtubules go, and they add a considerable amount of heterogeneity to the microtubules. So this is a microtubule over here, and you see you have the alpha and beta subunit of tubulins, and you see like along in a line the ones that are labeled, that's what we call the protofilament, and these assemble, and you can have a different number of protofilaments in every cycle and so on, and you make the microtubules. So a particular thing happens on the alpha subunit of tubulin, that if we focus on its C-terminus, it has a very characteristic tail of quite a few of glutamates. I'm sorry they're blue, they should have been red really. But you see like this is extremely glutamate rich, and there's a very characteristic tyrosine at the end. So one of the post-translational modifications happening in the tubulin tail is that it can get polyglutamylated. That's even more negative charges added. And this was something that actually has been recognized and sorted out quite in a short time. There's quite a few enzymes that do this job really. Interestingly, though, it has been realized very, very early on, in the 70s even, that the C-terminal tyrosine that is encoded by the tubulin genes, there are a few alpha tubulin variants, by the way, but it's being chopped off. That has been very clear. There has been antibodies papers 45 years ago. And what it also became clear is that this tyrosine can be religated, and actually that was sorted in the 80s, which are the enzymes that put back the tyrosine. But what had remained a mystery until 2017 actually, like 40 years after the description of detyrosination, is that what were the enzymes that were actually removing the tyrosine. Of course, the next question you can ask, is that important at all? Why do we care if the tyrosine is removed? And there's quite a few reasons to do that. There's quite good literature. This is characteristic, some characteristic papers that detyrosination is involved in cardiac function, detyrosination is involved in neuronal organization in the brain, and also it's involved in mitosis. So, and there's also quite a few other links to the neuropathy and so on. But all these were figured out indirectly with a few drugs that would inhibit detyrosination or enhance tyrosinated microtubules and so on. Nobody knew what were the enzymes. You could not genetically manipulate the system. So my colleague, group leader at our Department of Biochemistry at the Netherlands Cancer Institute, Tijn Brummelkamp, who likes solving mysteries using haploid genetics, started this quest like quite 6-7 years ago, actually 7-8 years ago. And what Tijn they do in his lab is that they have this beautiful system of human cells that are haploid. And it's important they're haploid because if they mutate them, by using a gene trap, for example. It's quite easy. If you get a single hit, you inactivate the gene. That's not necessarily the most physiological system ever. It's a weird cell line, but it's a fully functional cell line. It has all chromosomes, but it's haploid. So in that case, so what they do is that they mutagenize these cells with a gene trap they've developed that disrupt genes, and then they look for cells that, because of the mutagenesis, have changed their phenotype. And here, in this particular experiment, what they do is that they use an antibody that specifically recognizes detyrosinated tubulin or tyrosinated tubulin. And what you will see in this particular experiment, that you mutagenize and then use the antibody, on the top of this graph, you could see TTL, which was the known tyrosine ligase that was putting back the tyrosine, and there were a few hits below. And I will not go why these two were marked, but these were expected. They were not the detyrosinases, but related enzymes. But there was this hit for this gene called SVBP. And when we looked at the literature at this point, people have described SVBP as an extracellular small protein, a peptide really, it's only 40 amino acids, that was making a complex with some other proteins called vasohibins. Almost at the same time, there was this proposal based on bioinformatics that vasohibins might be proteases, so that filled the bill. And indeed, if you go, there are two homologs of vasohibins, VAS1 and VAS2. If you go in this particular cell line and you disrupt the vasohibin 1 and 2 genes, you see on the top line where detyrosinated tubulin antibodies. In normal cells, you have detyrosination. You disrupt the two genes, you have no detyrosination. You also see, like, if you stabilize microtubules using paclitaxel, one of these chemotherapeutics I mentioned, it doesn't make a difference. Well, like, it enhances detyrosination in normal cells, but in the absence of VAS1, it doesn't do anything. So that was, of course, very exciting, and a few years back, we determined the structure of VAS1 bound to this SVBP peptide, which was quite interesting structure because SVBP, at least the core of SVBP, is sort of inserted between the fold of VAS1, and it makes various specific contacts that are needed to stabilize the VAS1 enzyme. I'm not telling you the full story here because if you that was a trim domain that could crystallize, and if you go and take the full-length proteins and do some Sachs experiments of various constructs and so on, you realize there's an N-terminal disorder region, a C-terminal disorder region in both proteins, and if you can model them and start understanding things better. But this concept of disorder will come back a bit later. What we also did at this point is that if you look at the conservation color, to the orange is conserved, and you see also the electrostatics, you see this blue, and remember we had glutamates in the tail. It's becoming quite obvious where the glutamate tail is bound. And we have this beautiful program, HADOC, which what we like to it, it's not blind peptide binding, but we know the active site residues, we give it a few hints, then we dock the peptides, it gives us some ideas, we mutated more residues, we assessed them in cells for their function, and we took the mutants that were changing the phenotype, put them back to HADOC, and finally we got a rather convincing model of how the alpha tubulin tail is bound, which you see there in green. It is indeed bound in this open groove of VAS1. So now it's the time to admit that I have misled you a bit, because I showed you conveniently before the top experiment that we delete VAS1 and VAS2, and then you do not get detyrosination. However, if you go on different cell lines, what you find out is that even, so you see to the left, maybe you get rid of detyrosination in the absence of paclitaxel, but when you stabilize the microtubules with paclitaxel, then you see like you still get detyrosination, even if you're genetically ablated both genes from these cells. And you see that even in another cell line, this is even there without stabilizing the microtubule. So what does this tell us? It tells us that that's not the whole story. There is another detyrosinase as well, which has been hiding, and it did not come up in the screens. We looked quite a bit. So that's the point that a new postdoc in Tijn Broemelskamp lab, Lisa, we have 3 Lisas in the lab. This is getting so this is the typical blackout. So Lisa started these experiments, and what she did here is that she took the hub cells and she deleted the VAS1 and the VAS2 genes. And then she asked the question, like, if I go now and randomly mutagenize, do I get modulators of detyrosination? Of course, because now when you already get rid of VAS1, VAS2, it's not anymore a very easy task. There were a few other tricks done. One trick was to stabilize the microtubules with peptidactyl and look for positive regulators. And the other trick was also to get rid of the TTL, which puts back the tyrosine to enhance the detyrosination and also look at this case. So the consensus of that was a weird gene that I will say the name wrong. It was not even annotated as a protease, but conveniently we called it Matcap, which is microtubule protease. And it seems indeed to be the missing tubulin detyrosinase. I put a question mark here because actually there are 2 homologs of the gene. We worked on the one that we found. The other homolog, it's not very clear what it's doing, so I'll leave the question mark there for a while. So the difference here is that I'm showing the same experiment to the left with detyrosination, and you see when you get rid of Matcap on top of VAS1 and VAS2, then in every single cell line that we looked at, there is no detyrosination left really. And of course, when you take genes with VAS1, VAS2 that are Matcap depleted, and you add and you overexpress Matcap on these cells, you can reinstall detyrosination. That's what you see on the right panel. So that gave us a fantastic opportunity to get from a group in France, from Marie-Jo Moutin, the SVBP knockout mice and cross them with the knockout mice that were created in Tijn's group, and you see like progressively the double knockout of SVBP and Matcap. When you knock out SVBP, it's a trick for avoiding knocking out 2 genes in VAS1 and VAS2. You see the brain of these mice is getting smaller, and not only the number of cells in the brain is getting less, and also the weight of the brain is getting smaller. And actually, kindly, one referee asked us, like, so why does this happen? And that was the last panel of the last experiment, of the last figure of the paper, that this is in mouse embryos at 14.5 days, that you see in the subventricular zone, there are some progenitor, some stem cells that seem to be less in the knockout. So there's some problem in brain development that results in this phenotype. This phenotype, actually, I love to show this figure. There's a very silly test that you can do with mice. You give them some paper, like you give in your hamsters at home, and rodents like to make nests, right? You know, even for the kids or for their own and so on. So in that case, you see, like, if you take wild-type mice and you give them some paper, they will make nests. These are on top the scoring that we have for nests. So you see in the graph, the wild-type make nests. The Matcap knockouts, well, you know, a few of them stop making. To make a long story short, if you take the full detyrosination knockout, you see the mice, they do not like to make this nest. The females do not take care of their children. These mice are viable, which was already in itself a huge surprise, because when you knock out the tyrosine religase, this is a lethal phenotype. So if you cannot put back the tyrosine, it's lethal. So now it's almost logical. When we cannot take it out, the mice are alive, but they have quite some problems. Anyway, back to Matcap. We determined the structure. I like to say that it is very likely the last structure we determined in my lab by experimental phasing. We did it with a mercury salt, the old-fashioned way, because this was in insect cells. We could not make selenomet. And it was extremely useful to get that structure and work on it for a year and a half, and show you the work that we did. And at the time we decided to write up the paper. AlphaFold was out, and it had perfectly predicted this protein, which is a zinc metalloprotease, and you can start to get the link with Ida's work from yesterday. The zinc was not in the AlphaFold model. So how you start little projects, putting ligands in things. Again, we went back to haddock. We liked this. We started mutating. Here, actually, it was quite tricky because the groove was not very clear. And you see in electrostatics, the groove seems to have a bifurcation. So we were never sure with haddock where the tail would go. Would it go from a bit top to bottom? Would it fork? Would it come from the left? And so on. And that was something we were wondering. But to make a long story short, various, 3 rounds of mutagenesis, assaying the activity in cells, we could get down to what are the residues that are recognizing the tubulin tail, and how the mechanism is, and so on. So the next question that became relevant at this point is like, how does Matcap bind to microtubules? And Yitzke, who's sitting right there in the audience, has tried for quite a while to get microtubules decorated with Matcap, and has failed for quite a while to do it. And then at some points he had this quite desperate move that she changed the protocol with a double application, first write the microtubules and then put more protein, but at the same time she also used, together with Lisa, a cell line that was depleted in all the 3 detyrosination enzymes that we had in the meantime. So these microtubules were fully tyrosinated. And of course, we were using an inactive protein that still binds but cannot do the catalysis. And you can even see here in the micrographs, yes, not from a distance, but we could see all of a sudden changing all these things together. So we don't know what worked at the end, but all together at least they worked. You could see Matcap binding on the microtubules, and Yitzke had a lovely time computing for a while. And to make a long story short, we ended up with a structure that you see the alpha and the beta tubulin. It's a very good map. And then we could see Matcap. What you're also seeing, I guess, from this picture is that the resolution is not uniform at all. And the tubulin is better than 3 angstroms, closer to 2.8, I would say. And then you see the interface is quite well defined, which was good for us. And the other side of Matcap is at much worse resolution. Very likely there is some movement and so on. We did not do the sophisticated refinement and the different things yet, because we were focusing on analyzing that interface. But I guess we will. So a very exciting thing was that we could see the density for the tail, because when we docked the tubulin structures do not have the tail ordered. So when we docked our Matcap crystal structure to tubulin crystal structures in the EM density, we could see extra density, let's call it the difference in pink, that would fit excellent with one of the 2 haddock models. So it really helped us to see which of the 2 haddock models was correct. And also all the interactions that we were speculating to be there were confirmed. Now the interface of Matcap with the microtubules is really quite exciting because it's quite complex. So 1, it's the tail that binds on the active site, that's right in the middle. 2, there's 2 helices, the orange helix in the microtubules and the blue helix in Matcap that they interact. And when we go and we mutate residues there, we can alleviate function, so that's all is good. then you see 3, there is this little loop that is interacting with beta-tubulin. And that was very important for us because we had this experiment that we knew, and also the cell biology is telling you that Matcap is more efficient in detyrosinating microtubules and not tubulin dimers. So why does it recognize microtubules? We think that a determinant of that is actually this little loop that contacts not the, so the, in the middle is the alpha-beta dimer of the 2 tubulins. What you see below is the next beta of the alpha-beta dimer of the protofilament. So that's why we think it's one of the reasons that it prefers the protofilaments, because of this interaction. And indeed, if we mutate this interaction, again, we get a phenotype in cells, so this looks to be correct. There's also positively charged paths where you see the 4 in the back of the protein, that you also see under 4 in the other figure, that we see some density there on the maps, that we think that the C-terminus of the beta-tubulin might also be binding there, contributing to the recognition. And of course, the 5 is this floating 5, is that we still don't know what the role is of the N-terminal tail, that, however, we know that if we chop it off, we affect function but not affinity in the same way, which is also a very interesting puzzle for the future. So it's in the meantime, while we were doing that, there was another group that actually solved the VAS1 structure on microtubules, the VAS1-SBBP structure on microtubules. And it's fun because they had an easier time for their mechanism, because VAS1-SBBP, which also refers microtubules, is binding in the groove between 2 neighboring protofilaments. So it was immediately obvious why VAS1-SBBP likes microtubules. Matcap, you see, it really binds on one protofilament. So they both seem to like the microtubules, but one binding between protofilaments and the other one binding along the dimers of the protofilament. That's it. And what's next? It's quite stunning that nature came up with 2 distinct evolutionary solutions for the same problem: a cysteine carboxybeptidase and a zinc-dependent metalloprotease. They're really completely different proteins evolutionally. Actually, interesting, one of the closest homologs of Matcap is the ACE2 receptor of COVID. So now I have a paper that says SARS-CoV-2 as well, so it's legitimate. So they have different enzymatic mechanisms, as you can imagine. They bind in a different way to microtubules, so that leads to a few questions. They must have some kind of specialization. Is that on regulation in the cellular level? The different genes are regulated differentially in different organs or in different situations? Or actually, do they prefer different microtubules as substrates? And I'm sure you remember my first slide that I showed you the polyglutamylation tail. That is another modification nearby. So could it be that Matcap acts on polyglutamylated microtubules and VAS1 not, or the other way around? Open questions. We also know that detyrosination is required for brain formation, but brain was the obvious phenotype, get it done. What's the involvement in cardiac function? What happens if you if you treat mice with chemotherapeutics and check neuropathy? Are knockout mice not susceptible to paclitaxel-induced neuropathy? And of course, that brings to the question like, are these enzymes perhaps good targets for cancer patients that receive this kind of chemo and develop neuropathy? At least there's one company that they think it's a good target, so that's good. Anyway, so last but not least, all the curiosity and the cell biology was of course driven by Tijn Broemelkamp, who works on a variety of questions, but he does have this affinity for cytoskeleton. He also likes to see how the actin N-terminus is modified, but I'm not allowed to say much about that. Joppe Neuhaus was the PhD student that discovered SVBP, and Nassos Adamopoulos was a PhD student in my lab that did the VAS1 SVBP structure and the biochemistry together with Nassos. Lisa Langstrom was a postdoc that joined Tijn's lab after Joppe was leaving. She teamed up with Nassos for the mutagenic analysis of VAS1 SVBP, and she worked, of course, very, very close to Jitske, that did the cryo-EM, the crystal structure refinement, and collaborated quite a lot with Lisa over the past few months. All these papers have been published. Actually, it's a print version of Science yesterday that has the story of Matcap on. It was online for a while. And I would like to thank NEMI. This is the EM infrastructure in Holland. NASEN, of course, that we use for data collection. And it's being super useful complementation to our own gel with a K2 at the NKI that we're happy to have. NVO that will be funding the project from now on, actually, and on Code Institute and the Netherlands Cancer Institute that supported both Tijn's group and my group for all these years. So thank you. I'm sorry I made up for the time before. Thank you.